It's my pleasure to introduce our speaker today. As many of you know, Angela has been in the division of blood marrow transplant for a number of years. We won't say how many. And she is a professor of pediatrics in the division of bone marrow transplantation, and certainly she's here today as part of the TBT program within the Cancer Center. Her topic today is on the 3D biofabrication for testable cancer modeling, which I think is an important new novel strategy for looking at ways of evaluating either metastasis or looking at ways for delivering new drugs and seeing what the impact is in a 3D model system more closely resembling what happens in vivo. And as you also know, that Angela has been very instrumental and is now, I guess, the director of the 3D printing facility. Has been important, which I think you developed in about 2015, 2016 range. And certainly, I think many other institutions around the country have adopted the same type of modeling systems because of its potential impact in the care of the patients we take care of. Well, anyway, it's my great pleasure to introduce Angela Mortari, Panoskaltsis. Sorry.
Close enough.
Anyway, thank you so much for being here, and Angela.
Okay, great. Thanks, John. So thank you all for coming. And first, I just have a disclosure that there is a patent pending on some of the work that I will be presenting today. So first, in order to get to the point of why you're all here, we first need to go over a bit of background of what bioprinting is and all of the different components that are required to at least understand in order to start to biofabricate any type of tissue. And so what is 3D bioprinting? So as just written there, it's an automated type of layered manufacturing, except that it involves biologically relevant material. So it's not, I mean, it's like 3D printing, right? So from what you've all heard about, like, for example, I could, 3D print this, right? Using either plastics or metals, a pen or any type of object. But that is not bioprinting. So bioprinting refers to any time you're using some type of biologically relevant material, and it could be with or without living cells. So a lot of people, though, tend to conflate 3D printing and 3D bioprinting. And that especially is true for when 3D printing is done for a medical purpose. So for example, last year I had a hip replacement, and the acetabular cup in my hip was 3D printed. But that's made out of metal, and that's 3D printing, not bioprinting, okay? But it's for a medical purpose. So this field actually started a little over, I guess it's been 35 years now, by Mr. Charles Hall, when he developed the process of stereolithography in order to do like rapid prototyping of parts. And he thought about doing this while he was coding kitchen counters, actually, thinking of how to automate something like that. And then rapid prototyping, as I said, is used in many other fields. You can make, jewelry and shoes, dental parts. But the first use of a 3D printed material for medical use was actually a metal jawbone. But again, that's not bioprinting. The first demonstration of actually being able to print with living material was done here by our own Dr. David Odie in biomedical engineering at the University of Minnesota. And that was 20 years ago. And he used laser guided direct writing. So it's a bit like if anybody's done laser capture microdissection, this is like the reverse, where you're putting cells onto a substrate. And he could demonstrate precise microscale patterning of viable cells. But he did this in 2D. And other people since then have used this technology in order to do 3D printing. And the way that it works, at least with this methodology, is that you have, your substrate at the bottom here that you want to print something onto. And then up here where you have your target, you can use, for example, a piece of quartz or a glass slide. And that slide here in gray would be coated with something that can absorb the power of the laser. Otherwise, your cells are going to get fried. So, for example, we use gelatin for that. And then you can have a gel here that contains the cells of interest that you want to transfer. And when the laser shines on here, this power absorbing layer ends up causing this bubble to form, and that induces forward transfer of the cells onto the substrate. And that's been done by now several people, not just in the US, but in Europe too. So that's one form of bioprinting. Another fact that is interesting to remember is that cells actually like to self-assemble, and they stick together. And this gentleman here, Gabor Forgash, realized over 20 years ago that when he was watching embryos form in chick eggs, that the cells tend to move together like a fluid. And so he reasoned from that you should be able to actually use an extrusion type of printing method, an automated method, in order to print cells. Oh, and there's Gabor and me. And then he has also since then demonstrated that you could even do scaffold-free tissue engineering because of this cell self-assembly characteristic of cells. So you could actually engineer tissues with cells only and not even using a scaffold with the cells. However, he did use some what we call like support materials in order to it's like a sacrificial layers that you could then remove after you've printed the cells of interest and the positions that you need. So here's just a demonstration showing that if you're printing spheroids of cells or clusters of cells, you could print them in this type of layered manner and then get rid of the sacrificial material. Then you're left with, for example, here he was trying to show that, bless you, he was trying to show that you could make a vessel. The first bioprinters were actually made back in 2004, so it's not that long ago. This is a really recent technology. The first two people in the same year was Wei Sun at Drexel, and this is Tom Bowen here. What he did was he just took a regular inkjet printer out of the office, and he adjusted the heads, the cartridges, so that they wouldn't fry the cells, so lowered the temperature on them, and he was able to print cells with pretty good precision. And that's some of us there. Just showing you that people that bioprint do have fun too. So what is the purpose of bioprinting? So the obvious use that most people think of, when you tell even the lay public about 3D bioprinting, they'll think of making a tissue or an organ for transplantation purposes. And yes, that's true, and that's probably a little more distant in the future, although there are some cases that I will describe briefly of where something actually is being used in man. But also to make small three-dimensional models for drug screening purposes, and even just to study cell behavior. Because most of what we know in the textbooks, right, about how cells behave is done on hard 2D plastic, cells grown in tissue culture. That is not at all a normal environment for a cell. That's a really tough and hard environment. Basically the cells are laying there naked, they're spread out, like this. And for those of you who can't see this, I'm just, spreading my arms out because they're trying to touch their nearest neighbors and create a microenvironment for themselves. So it'd be like, taking us and stripping us of everything that we have in our clothes and throwing us up in the tundra and expecting us to exhibit normal human behavior. Well, we'll exhibit some behaviors that you might see in your normal environment here in the city, but certainly not everything. Another example is to print actually medications, pills. So there are now poly pills being created that you can put in multiple drugs into one pill. In fact, FDA's already been approving bioprinted pills. And even if it's a drug that's already been used and FDA cleared previously, the fact that it's bioprinted, it needed to go through FDA approval again. And also scaffolds to guide tissue growth. So not printing the tissue per se, but to print a biodegradable scaffold in order to instruct the tissue in what direction to grow into. So for example, this is being used now for helping to guide nerves to grow for peripheral nerve injury, being also experimented with for spinal cord injury. Also has been used for example, bronchomalacia, where there's complete collapse of the bronchus. You can build a scaffold to hold it open as a splint, as opposed to putting a stent inside where it can cause even more injury. So these are all other examples. And the thing too is that you would want that scaffold to then biodegrade over time so that you wouldn't have to go back in and remove it. And this is what's being done. And in fact, it's already been done in people. So creating organs, as we said, the most common reason is for transplantation purposes. But drug screening and testing. And the easiest tissues to make are those that are thin or avascular, which means they don't need a blood vessel supply. So for example, parts of the eyes, like the cornea, the lens, the retina. In fact, corneas are already going into man, and retinas as well. Cartilage, already going into man in experimental cases. Let's see, thin skin samples. Those are other things that are being made. So those are the easiest to do, because one of the major challenges right now in bioprinting is that we are limited by how thick we can build a tissue. Because if it doesn't have enough of a blood supply to get oxygen in and nutrients out, then everything's going to die if there's not enough oxygen tension in there. So because in the body, those cells that need oxygen are usually never further than 200 microns away from a capillary, at least. And so that's been the real challenge in bioprinting, is how to get those vessels established fast enough so the cells can make it. And it really requires a multidisciplinary approach. So for example, in my lab, there's not just stem cell scientists, but there are physiologists even, but there's a lot of engineers in there, people that know material science, mechanical engineers, computer scientists, we have surgeons. So it's just a huge interdisciplinary group, which makes it a lot of fun too. So of course, as you can imagine, using organs to make organs for transplantation, you could, exploit the use of iPS cells or other types of progenitor cells in order to create an organ that can be made out of the patient's own cells, so it wouldn't be rejected by their immune system. So we could either use histocompatible off-the-shelf cells or, make their own. Now, for drug toxicity purposes or for drug screening, you could use generic off-the-shelf cells that are well characterized and standardized cell lots. But for drug efficacy, you could still do that, but you might also want to use the patient's own cells. So for example, if it's, the patient's, has some type of mutation that causes a pathology, you might want to test it, and it's a rare disease maybe, you might want to test it on their own cells. Or if it's their own tumor, you might want to create the model using their own tumor cells so you can see what combination of drugs or cell therapies would work best for that person. So what do we need in order to print a tissue or to biofabricate any tissue? So, ideally, you're going to need a scaffold or the extracellular matrix, either at the beginning or rely on the cells to make their own matrix. Of course, you need the right cells to put in. At what stage of differentiation should you put them in? That will depend on the tissue that you're making. A way to get nutrients in and waste out, a way to measure the function, and preferably non-invasively, because if you have to go in and do, a biopsy or destroy the tissue in order to figure out what you made, then you no longer have the tissue. So there's a lot of work being done right now by other types of engineers trying to make inline sensors and monitors to figure out the functionality of the tissues that are being produced. And so just to drive home the point of what the extracellular matrix is, you've all heard about decellularization. So this is an example from my lab where we've taken a set of pig lungs and decellularized them with a series of detergents. And what remains is that extracellular matrix. And just so for students who've never seen this before, I think I have to disable this laser pointer first. And then here's a decellularized lung that is now connected to a ventilator. And you can see that it inflates. And no, it's not breathing. There are no cells in there. It's just being ventilated. But, isn't that amazing? Just, how tightly meshed the matrix is. It doesn't even allow air to escape. So that's just, so that to drive home what the matrix is. Okay, if you don't take anything else home from this talk today, is what is the matrix? Okay, so it's mostly composed, of course, of collagen, elastins, and dozens of other proteins. So other things we need to consider in bioprinting is, what is the function we're trying to make out of the organ? And maybe you don't need, all the function, but you need just enough in order to demonstrate what you need to establish with maybe the drug that you're trying to experiment with or to validate. Or it might be a tissue for a patient, and it's just, it just needs to have enough of a function in order to improve that person's quality of life. It doesn't need to do everything that the organ usually does in the body. Need to figure out a way to get nutrients in and waste out. So for example, you might need to, create a vessel system inside the tissue, so then you need endothelial cells to line that. And also, especially if you're using this for transplant purposes, you need to figure out a way to maintain the organ and have self-renewal properties. Because if it's not going to self-renew, then there's no point doing it. Because then just be back to square one within weeks, probably. And also, you need to think about what other cells need to be incorporated within your construct that can help maintain the environment and maintain the matrix. So other things are cell culture scale-up. So because there are going to be different cell types involved, you need to really consider the timing and the derivation and the cell numbers that you need and at what stage of differentiation you're going to be using them at. And also, you need to think about when you're bioprinting anything, even a tumor, how long will the cells remain viable and functional? And that you have to link with how long will the hydrogel that you're using for printing, and we'll talk about hydrogels in a minute, but how long will it be printable? Because sometimes they're only printable for about 30 minutes. So can you get enough of your construct made within 30 minutes? Other gels, are printable for much longer periods of time. And also, if it's taking too long to print your construct because maybe it's too large, will your cells remain viable while they're sitting in the cartridge and the syringe being extruded for printing purposes? So then you might need to think about temperature and other types of nutrients that need to be immediately, start getting perfused within the construct in order to keep things alive. So then the functional readout, how much do we need to know? So for example, the FDA, might not be interested in the 1,000 things that you're measuring. They might say, we only care about these 8 things. So, that's something that you would need to establish with the FDA beforehand to say, so that you can then design your experiments appropriately and have the correct readouts that you need. You might integrate some surrogate markers. So for example, we use a lot of fluorescent tags. Or you might use some small molecule chemistry that when cells, one cell type makes one type of chemical or molecule, another cell makes another type, and they combine together, it creates something, some color change or a fluorescence to happen. Or you might have, your functional readout based on a promoter that then drives expression of another fluorescent protein. So then you know that your cell is now producing that product. Also, you have to think about, okay, does my construct need a bioreactor? If it does, do we have one? If we don't have one, then we probably need to build one. So then you got to call in engineers to start helping you build bioreactors because biologists aren't going to do that, most likely. So here's some example of tissues and other things that have been, oh, I think I forgot my laser here. There we go. That have been biofabricated. So skin, because it's thin, it's much easier to produce without relying on a big vascular supply. Cartilage, vasculature here, aortic valve, the tracheal splint, talked about that before. And then here it's just showing in the future, a solid organ, for example, a kidney. No one has yet bioprinted a kidney, okay? No one's bioprinted a liver. No one's there's just so many of these organs that people are aspiring to do, but a whole intact organ has not yet been bioprinted. So people are exploring ideas to maybe, for example, in the kidney is maybe to print small parts of the kidney and then put them together like Legos. That might be one approach because then again you're relying on the self-assembly characteristics of cells in order to do that. But people are starting to make some progress. So the 3 main steps in bioprinting, of course, is just like any engineering process. So there's a pre-processing stage where you have your design, your CAD model, and some of that could be based off of images from an MRI or CT. So you can get your 3D files from that, your information. Growing up all your cells and the bioink prep. So growing up the cells is probably the most time-intensive and labor-intensive and expensive part of this. The processing itself, which is the actual printing, does not take long at all. And then post-processing, of course, you need to think about, do I need to perfuse the tissue? Do I need to allow it to mature? Or am I going to implant it and allow the body to be the bioreactor and allow it to mature there? And also the monitoring, how are you going to monitor it, hopefully non-invasively after printing. So bioink, just like when you have ink for your printer in your office, we have bioink. So that's the raw material of bioprinting. So they're really soft materials. They can be with or without cells. And so the ones that are considered successful are the ones that can sustain cells, and also that preferably have adhesion sites for integrins to bind to, so that it allows for cell spreading and proliferation, and also allowing for extracellular matrix interaction. So again, we talked a little bit about the bioprintability. So for example, like an ideal hydrogel would have the consistency of like thick honey. So that's like, it feels like you're printing with honey when you see it print. So these are, some of the rheological properties. What is the viscosity of this solution that you're making? Because if it's too thick and too hard to extrude, then you might just blow up your printer. That would not be good. So preferably have some biomimicry in there. Have enough of mechanical integrity and stability so that when you print, it doesn't just, fall apart on you. Also, it should be insoluble in cell culture medium. Okay, because if all of a sudden, you print something, you put it in culture medium, and then it dissolves, well, that's no good. Now your construct is gone. Preferably should be biodegradable at an appropriate rate, especially if it's going to be used in vivo. And then some of the obvious things, non-toxic, non-immunogenic, and promote cell adhesion, which we talked about. So the two most common types of bioinks are the scaffold-based, that's very common using hydrogels, and we use a lot of decellularized extracellular matrix components too from different organs, because our reasoning is that if we're using the extracellular matrix from that organ, then the cells are more likely to behave like they should be part of that organ. So we have people in the lab working on making trachea, esophagus, skin, and vessels. So we've been decellularizing all of these different types of tissues. And then they're scaffold-free, relying on just cell self-aggregation. So what are hydrogels? They're really polymers, and they get cross-linked. They absorb a lot of water, up to 1,000 times their original weight, and they're very permeable. And some of the main groups are the naturally derived and the synthetic. So gelatin is very commonly used, and so is fibrin, which comes from human plasma sources. Collagen, chitosan coming from the shells of the outer coatings of insects. Alginate from algae, and then there's some synthetic hydrogels too that I'll talk about later. What's important, though, is that when these, the ideal hydrogel would exhibit a property known as shear thinning. So any of you who have tried to take ketchup out of a bottle, and you're shaking it, you're hitting it on the back, and all of a sudden it just splurts out all over the place, that's shear thinning. Okay, congratulations, you've experienced shear thinning. And then when it gets on your plate, it solidifies again, doesn't it? At least it congeals again. So basically, what happens in the hydrogel is that the polymer chains, as it's being extruded through the nozzle, become aligned. And so that, as you're applying force, so that's shear thinning. Shear thickening as you apply force is called rheopectic behavior. And that would be like, for example, if any of you have made mixed antigens with adjuvant, like alum, and you go back and forth between the two syringes and it gets thicker and thicker, you're making that emulsion. So that's rheopectic behavior. That's shear thickening. So I'm just going to talk about two hydrogels that are very commonly used in bioprinting, and they are also used in a lot of the work that we do in the lab. One is to use gelatin, which is just degraded collagen, but it's methacrylated. And the methacryl groups are added to the amine side groups of the collagen pieces. And so what this does is it allows for cross-linking when you use a photoinitiator. So when you can just cross-link it with blue light. And we do that a lot in the lab. And in fact, we make a lot of our own methacrylated gelatin, and it's available to you as well in the bioprinting facility. So a very common photoinitiator we use is this thing here called LAP, and you can just see that this has a very long name, so I'm not going to say it. But it allows for the cross-linking of those methacryl groups that were put on the amines, and then you get cross-linking of those polymer chains. Pluronic is a very, very interesting hydrogel, and it's a synthetic, and there are many different kinds, but it's something that undergoes a process called reverse gelation. It's a very interesting property. So how many of you have made Jell-O here? Many people have made Jell-O. So you take the powder, you dissolve it in warm water, right? It dissolves. You put it in the fridge, and what happens? It gels. It gels. So Pluronic has the consistency of, say, toothpaste coming out of a tube. Okay, so you print with that at room temp, and you put it in the fridge, and it turns to a liquid. Okay, so it exhibits the reverse property. But we can exploit this property in a very interesting way. So if you're printing some matrix material, say, and then you print a little squiggle line of this Pluronic stuff, then you just finish off your construct by printing more of your other collagen, let's say, on top. Then you put this construct that's all gelled, and you put it all in the fridge. And then what will happen is that Pluronic will turn into a liquid, and then you can just flush it out. And now you have an empty channel inside this gel construct. And then what you can do is now coat this channel with, say, endothelial cells to make a mimic of a blood vessel. Or you can coat it with epithelial cells to create a renal tubule, or whatever it is you're trying to make. So there are some very interesting properties of hydrogels that we can exploit in many different ways. Different types of bioprinting methods. There's droplet inkjet type printing, extrusion-based printing, which is the most common, laser-based printing, which I described already, biopaper-based, which is basically taking some sheets of polymers, printing on each sheet, and then piling them one on top of the other. Then you end up with your 3D construct. 2-photon polymerization, you can get some very nice resolution down to 1 micron resolution with that. These other ones, you get to about, with extrusion and droplet printing, you get about 80 to 100 micron resolution. And for a tissue, that might be okay, right? Because you're printing many cells at once. Laser-based is accurate to a single cell level. So basically 10 micron resolution, because you can print, 1 cell at a time, and you can print thousands of cells per second, depending on the frequency of your laser. Acoustophoretic, that's using ultrasound in order to get cells to align in different spots, depending on where you've put your plates that are emitting the frequencies. So that's another very cool method. These are just showing you examples of how the techniques work. So here's an example of an extrusion type process, just laying down the base right now. And let's see, can you see what this is? Who can see what this is? What is it? Yeah, an ear. Very good. Cartilage for an ear. Awesome. All right. So, and then here's an example just showing, the type of resolution we can get with printing by extrusion. And here is that example I was telling you about in using Pluronic to create a blood vessel mimic system inside a gel. And this was done by Jennifer Lewis's lab at Harvard. And so here you can see that they've lined it with endothelial cells, and here's a cross section showing you that there's an empty lumen inside. Here's another example of bioprinting, but this is now, instead of printing in air, this is printing inside a slurry or a suspended hydrogel. This is called free-form reversible embedding of suspended hydrogels. And they called it this so they could use the word fresh. Okay. And this was done by Adam Feinberg's lab. In fact, he's coming here to give a keynote in April at a Biofabrication conference that I organize every year. So basically what you do here is you have a slurry bath, a support bath, and then you print inside it. And this is really good for constructs that might be very fragile and would otherwise collapse on themselves if you're trying to print them in air or wouldn't have enough integrity. And so this slurry works like that hand gel material you've probably heard of called Purell. And you notice how there are bubbles inside it and they never float to the top. It's because it's not a liquid, it's microparticles. And so that's why this works. So it's basically made out of gelatin microparticles, and we also produce this in the lab for any of you who are interested. And you can get some pretty nice resolution with this too. And then what you can do is just, using temperature, then melt away all of the support part, and then you're left with your construct. So here's some examples. In my lab, in the bioprinting facility, I should say, this is using a 3-head printer. Not only can it print hydrogels, but you can do thermoplastics and other types of materials all with the same printer. So that's just showing extrusion printing. This one here is showing you the embedded printing. You can see the needle inside there. And next time I'm going to tell my student to use some food coloring so you can actually see what's in there. But you can see the whiter stuff in there is being printed inside the less whiter stuff. Okay. Oops. There we go. Okay. So here's another example using a 2-color print, showing that you can print different patterns, one on top of the other, on different materials. Here's an example of skin that was printed with laser-assisted bioprinting. This was done by a group in Germany several years ago now. So they printed, I think it was probably 40 layers. So they printed 20 layers of fibroblasts, 20 layers of keratinocytes, and then implanted it onto a mouse. And then you could see after 11 days, it got vascularized by the host. In situ bioprinting. So this is very interesting. This is also done by laser-assisted bioprinting. So here's an example where they had taken a rat and created a skull defect, and then they had anesthetize the rat, obviously. And while it was still alive, have it on the stage, and then they, using a laser-assisted bioprinting, printed bone material right on the skull while it was alive. So the idea later would be, and especially the military is interested in this, for people out in the field who've had bad burns, chemical burns, whatever, or bad injuries. You could, for example, if it's on your arm, put your arm in a scanner, get it scanned, outline the areas that need to be replaced, and then using the person's own cells, then print directly on their skin. Some interesting nozzle designs are important to consider when you want to bioprint anything. And that is also determined by the gels that you're going to use. So for example, you might have a thermosensitive type of hydrogel that requires to be printed at a cool temperature, and then it congeals when it's at a warm temperature. So you might need a cooling jacket on the cartridge in which your cells are located, or your gel's located. Or you might be using an alginate. An alginate is a hydrogel that is cross-linked with calcium. And so you might need to have a design inside your nozzle and your cartridge that keeps those two things separate until they meet at the point of print. Because if they were mixed together within the cartridge, it would just gel in there and cross-link, and then it would just, the pressure would build up so much, and again, you have a blown-up printer. Or it might be photosensitive, and you need to have a laser or some other type of light source shining right at the point of print so that it gets cross-linked immediately upon printing. Here's another example of using a coaxial nozzle. So where you're trying to print perhaps a tube, and you want to have those two things separate until they meet after extrusion. So it's like having a tube within a tube. And then when they come out, they get cross-linked. There's also a biopen that's been made by a group in Australia at University of Wollongong, and I like saying that word, Wollongong. So cool. In fact, the next biofabrication meeting next year is going to be in Australia. So what they've done here is, so they've created a handheld device that you could bioprint directly, and so they've been experimenting with this in pigs, where the surgeons are correcting vascular defects, heart defects, for example, by printing cells directly during surgery. So this would really revolutionize that field too. Here's another example from Ali Husseini's group, who's now at UCLA, where he's figured out a way to get multiple components all to come together and out of one extrusion nozzle. And he's been able to automate this and have it computer controlled so that he can control which components are coming out when and for how long. So he can create mixes of these hydrogels, that was really amazing when that happened. So here's just, again, examples, just using hydrogels, no cells here, but just showing the diversity of constructs that can be made and using, different combinations of different gels. Oh, and there's me and Ali. Okay. So here's an example from Jennifer Lewis's group where they made basically a mimic of a nephron. So they're trying to create the renal proximal tubule, again, by that method I described before, using pluronic. And then coating this now, though, with renal epithelial cells. And then, in fact, what they did was, and then here's just a cross-section. Oops, again, I don't have that pointer showing. Here we go. The cross-section across the construct here, and then here's showing all the open lumens across this entire tubule. And then what they did was they, in fact, exposed these constructs to varying levels of cyclosporin, which we know would be toxic, and demonstrated in a dose-dependent manner the toxicity. So they basically have established this as a drug toxicity model. Here's another example showing trying to print, more complicated constructs. So 2x3s, 3x3 designs, as you can see, layering one on top of the other and having all of these able to have empty lumens and perfuse cells through them. But what do you think is wrong with this construct? What's wrong with this design? These are cell biologists here. Come on. What's wrong with this design? It's square, yeah. There's nothing in your body that's at right angles, okay? We'd look pretty weird. Okay. So all of your vessels bifurcate at 45-degree angles. And so when you have cells going around corners like this, it creates a lot of shear stress. And so that's going to injure a lot of cells. So you need to think about these things. And, engineers like to do this stuff, but they need to talk to biologists. So, yeah, so I'm just like, good try. All right, so now let's get to why you're really here. And that's to talk about 3D bioprinting of an in vitro metastatic cancer model. This was work done by Fan Ben Meng, who's a postdoc in the lab. He has a PhD in materials science. And so we really had to teach him a lot about cancer and cell biology. And he's just done a fantastic job on this. And he is also in the lab of Mike McAlpine, who's a professor in mechanical engineering. He came here from Princeton about 3 years ago. And so he and I co-mentor Fan Ben on this project. So just, for this audience, just to refresh, we don't need to really go over it, but just that tumors aren't simply clones of cancer cells, right? They're composed of many other cell types, and even the cancer cells in there are heterogeneous. But we have to think about, in creating a tumor model, it's not enough just to create a tumor spheroid. But we have to think about, that it has a blood vessel supply. It has fibroblasts in it. It has all kinds of other vessels and other types of supporting cells. So it's not just cancer cells. Because when we're trying to study, how a drug will interact or affect a cancer, a tumor, or cell therapy, we also have to think about all of this crosstalk that's going on with all the other cell types inside that tumor. It's not just about the cancer cells. And so ideally, you'd want to reconstruct an in vitro environment that's pretty dynamic and consistent with trying to remodel all that extracellular and intracellular material, at all of these levels. And so the challenge is how are we going to establish precise placement of all of these critical components based on the physiological functions of all of these different cell types. And also, how can you create this dynamic environment, chemical environment, using multiple molecular factors? And so what we did was use a technology that had been previously developed in Mike McAlpine's lab with Fan Ben back at Princeton, and that was to use these programmable release capsules. So basically, they're capsules that have a shell, and I'll describe them to you in a minute, but you can have a cargo inside them. And they can be printed with very high precision. You can get pretty homogeneous sizes of these capsules. You can tune their size by just adding, more droplets in one spot. You can do this in nanoliter quantities. And you can get, different combinations of drops, within one drop in order to create certain chemical or biochemical reactions. And so basically, you can have a cargo inside, and then it's encapsulated with a polymer. And this polymer can also be imbued with gold nanorods. And because of the length of the gold nanorod, you can expose them to a laser of a wavelength that's just off of the length of this nanorod. And it causes a plasmonic resonance. And when that happens, then the capsule bursts open and releases the contents. And you can do this in a programmable way. And how do I mean by programmable? It's because you can get gold nanorods of different sizes and different aspect ratios. And so here, for example, is showing the creation of 2 different types of capsules using 2 different sizes of nanorods. So one capsule type is coated only with nanorods of 650 nanometer lengths, the other ones of 785 nanometer lengths. Now, when you have the laser exposed to a 783, for example, nanometer laser exposed to gold nanorods of 658, of 650 size, say, they will not be triggered. Whereas if you expose that laser to capsules that were coated with gold nanorods of 785 nanometer length, then they are released. So you could even have them within the same construct. And if the laser that you're using would only trigger the one type of capsule, then only those would get triggered. And then if you wanted the other cargo to be released later, you can then use another laser wavelength. So that's what we mean by programmable. So here's for an example, just showing that you could, do a lot of this droplet printing, within constructs and create some pretty complicated patterns with them using two different, this is just using food coloring for now, just for demonstration purposes. But you can get some pretty complex arrays created inside three-dimensional hydrogel matrices. And then here's a printer that Fan Ben built, and this was what was used to create the metastatic cancer model that I'm going to tell you in just a second about. And what's important about this is that all of the components that were printed were all done using the same printer. So not only did we print the chamber with silicone, okay, using this printer, but the cells were also printed, and the matrix material was also printed using this printer. So you could load this with various cartridges across the top here, and then have a laser shining on it, and then you can, do all of this in an automated way. So, so the idea is that in order to create this dynamic chemical environment, we would have capsules that are coated with gold nanorods containing some type of material that would guide the migration of cells, because that material would, bind to the surface receptor of the cells. And so here was our design. So we had, for example, created this main blood channel here, blood vessel channel. And then on this side, we had capsules containing VEGF that would guide the sprouting of blood vessels in this direction. And on this side, we had capsules containing EGF that would guide the migration of cancer cells in the opposite direction. And so the capsules closest to the blood vessels were triggered the day of print. And then 2 days later, we would do the next row of capsules. And 2 days after that, we would do the next row of capsules. And slowly, we were creating this chemical gradient that was more reminiscent of what happens in the body, as opposed to just flooding the system with a growth factor and confusing the cells about which way to go. And so then here, we print the droplet of the tumor cells that we're interested in. And we've done this with many different types of cancer cells. So for example, using epidermal growth factor, that we had that was Texas red labeled, and we could show that after triggering with the laser, you could get release of this fluorescent growth factor over time. So over 2 days after this one spot, it would all be gone. So here's just showing the kinetics of the release. So if you just have a bare core with no shell, this is its immediate release of the material into the gel. If you have the capsule with a laser, this is the nice slow release that you get. And then without the laser, that's what you get. It's just very, very minimal, very slow type of leak here. And then here's where we've done one capsule here, laser triggered it, and then we had a collection chamber on this side of the gel and a collection chamber on this side of the gel, and showed that on this side, there was a chemical gradient being formed. So there was more chemical released on this side than there was out here. So you could actually make a chemical gradient. So now to guide tumor cell migration, we just demonstrated this very simply by printing droplets of cancer cells and then 3D printing also the capsules. So you 3D print the core, you 3D print the shell on top containing the gold nanorods. So the core contains your growth factor. Then triggering release, and on the other side, we would have control capsules just containing just the gelatin core. And then here is the silicone chamber that is printed, 3D printed as well, so that's around here. And then here you could put, in fact, up to, you could really scale up this whole process. So here you could print, for example, 5 droplets of the same cancer cell type or 5 different cancer cell types. And then, look in the same system and see how they compare, and then coat the capsules on this side and this side. Now, the significance of these 3 prongs here it has nothing to do with the biology. It just has to do with keeping the gel intact and keeping the shape of the gel so it doesn't constrict or contract too much. And then here is just showing how these cells migrate in response over time. See the nice slow response over time? So this is up to day 10. And also, do you notice they're only going in one direction? They're not going the other way. So this is guided migration. And then you can track these cells. And then we were able to calculate that they were traveling at about 15 microns an hour, as opposed to cells going in the other direction that were barely moving, only 1 micron per hour. And then cell intensity, so there was accelerated proliferation too, so they weren't just migrating, they were also proliferating in response to the EGF. So this is with epidermal growth factor release, and this is without. And also, we could demonstrate selectivity. So even if we used, so the cancer cells that I just showed you were lung cancer cells, the lung adenocarcinoma, that's the A549 cell line. We also tested this with melanoma. So you could see here, this is the cell distribution. So on day 0 and day 10, how much. And then even with a melanoma line that's very aggressive, you could still guide the migration in one direction. So it's pretty amazing. And then these are just human umbilical vein endothelial cells, and they don't migrate in response to the EGF. So it's just showing selectivity. Now, formation of the vasculature within this model. So we changed the design a little bit here. Here's the silicone chambers here. And what we did, so you can either create the main blood vessel channel with pluronic, that interesting hydrogel I told you about earlier, or you can just use a metal pin mold, which is very low tech, and it's just faster, so we just eliminated a step doing that. So just showing that we can, perfuse things through it, doesn't leak out, so it's an intact vessel, has a lumen. And then here's showing after release of capsules containing VEGF that, in fact, the cells, the vessel is sprouting in only one direction, as you can see. So if you just have VEGF all over in the hydrogel, just contained with everywhere, mixed in with the hydrogel, these blood vessels will just sprout in random directions. They wouldn't be guided in one direction like this. And so then this is how this was set up. So here's the main vessel. Here's the printed tumor cells here. The VEGF capsules here. The EGF capsules here. These are laser triggered. And then you get cancer going one way, the vessel sprouting in the other direction. So here's what it looks like. And we also did this in the context of fibroblasts mixed in within this gel, which was fibrin, made of fibrinogen and thrombin. And then over time, this is what happens. So the cells start migrating toward the vessel starts sprouting towards the cancer cells. And the other thing that we inadvertently discovered was that we could also collect metastatic cells from this. So over time, in the chamber, so if you remember, here in this chamber, here, you see that we can screen drugs in through the chamber this way, and through this vessel. And then there's an outlet here. So in this chamber over here, we discovered that a lot of these cancer cells were coming out. And these were cells that self-selected to enter the blood vessel and migrate out this way. And we could collect them and demonstrate that they indeed expressed increased levels of markers that are known to be expressed higher on metastatic cells compared to those cells that were exposed to the same conditions with EGF but remained in the hydrogel. And the only way that these cells could get into this chamber is if they entered the vessel and went there. And then here's an example showing the effect of using these constructs as drug screening models using some immunotoxins that we got from Dan Valera. So he has made some with using Pseudomonas exotoxin, and he targeted with either EGF in order to target the EGF receptor on the cancer cells, or an off-target immunotoxin, anti-CD22, which should target B cells and not the lung cancer cells that are shown here. So you can see here with the immunotoxin that should kill the cancer cells, that it doesn't really doesn't allow for the cells to spread or to grow, and in fact they start to decrease over time. This is the off-target immunotoxin. The cells expand just as they did with no toxin. And here it's just showing the green channel, not showing all the red cells, so you can see how the tumor is growing or not. And then this is just showing the cell viability. So the cancer cells here determined by fluorescence intensity, showing that the EGF, in fact, is killing the cells, and the CD22 targeting immunotoxin does not. And then this is a very interesting graph here, and I think very important. So here in the black, it's showing you what happens when you expose these cancer cells to the immunotoxin in 2D tissue culture, which is typically what Dr. Valera does in the lab to determine, what a possible dose should be. But now here's what it is when you do it in our 3D model, but without fibroblasts in the gel mix. And then here is how the cells, the rate of death when you have fibroblasts in the gel mix. So you can see that it really slows down the killing of the cells, because those fibroblasts also act as a sink. And so this model actually demonstrates and mimics more what happens in the body when the immunotoxins aren't killing the cancer as fast as you thought they might have, just by doing them in 2D, looking at it in 2D. So basically just showing you, with the construct, how it was designed, showing you early on the sprouting of vessels towards the cancer cells, and then ultimately you get this vascularized tumor that you can screen with. And this was published earlier this year. And he has since now started to use some other interesting designs in order to maybe accelerate this. So he's in fact incorporating now some endothelial cells within the cancer droplet and getting these connections to form even sooner. You can see here he's infused some microbeads and starting to see them within the blood vessels that are connecting between the tumor and the main channel. And also this is within the tumor droplet. You're starting to see some very nice capillaries with lumens inside too. So this is the bioprinting facility. If any of you are interested, so start thinking about how you can utilize 3D modeling and bioprinting in your own work. Maybe it's not about cancer per se, but it could be about any other type of model system or cell model system. So these are printers that are used for experimentation as well as for training purposes and for classroom settings. So I teach a 3D bioprinting course every fall. That you can take. It's a 2-credit class. This is in fact the machine that Fanben built to do the cancer metastatic models. This is a laser-assisted bioprinter that we have in the lab. This is not a bioprinter. This is a fluorescence microscope with an on-stage incubator that you can control gases and temp, and so you can monitor your constructs over time without having to take them in and out of an incubator. This is a soft tissue mechanical testing device. You can test the stiffness of the material that you're building and see how well it approximates native tissue. And this is all available as an ISO, ESO. So if you'd like training, you just go to the website here. There's also a flyer over there you can look at. So this is just people printing in the lab. And I'd like to thank you. This is Haley here. She manages the bioprinting facility. So if you're interested in training, she'd be the one who would be training you. So I'd like to thank all these people and our funding. Thanks. I'll take questions now. Okay, Carol Lang has a question, and we're waiting for the microphone to come to Carol.
Thank you. So fascinating. So talk a little bit about feasibility. How expensive is one of those things? I mean, what kind of operating budget do you have? It sounds like you've blown up a few by the way you talk. Like how, like if I wanted to come do an experiment, what are we talking about?
Yeah, so these bioprinters here cost $6 an hour. Yeah. Training is, I believe training, it depends on how much training you want, because we'll even train people on cell culture, fluorescence we'll do all of that, because indeed there are people, like there have been dentists come, all kinds of people come for training, and from industry too, that need all of that. But if you just want to learn how to do the printing or maybe the CAD design, maybe just 2-3 hours. So it's about, I think it's about maybe 60, 62 an hour for the training part at an ISO rate. But some of these constructs take, it depends what you're making. Some might take 5 minutes to print. Some might take 20 minutes. So, yeah. And if you're printing with silicone, you can print the silicone part and then set it aside, because that doesn't matter, right? Just leave that and then print the rest inside it.
What if I don't feel comfortable learning how to use this equipment for fear I will blow it up? Mm-hmm. Do you have staff that yes. a project could be brought to?
Absolutely. We do that as well. Yeah. We do that as well. And the whole machine doesn't blow up, okay? It's just the cartridge, things will just pop. because usually the pressure that we use to get the extrusion, instead of using an air compressor, usually we just use a nitrogen gas tank because it's quieter and cheaper. And then what'll happen is where things have been connected, they'll just pop open. But yeah, occasionally there's been things just come all over the place. But,
so you have to do an indemnification contract
Yeah.
before you actually participate.
yeah, exactly.
So when you were showing the slide where you have the tumors cells migrating towards the EGF, have you had the opportunity to select out those cells and do it, sequence the cells, or to see what kind of expression profile that might be different in those that are moving than versus the bulk of the cells actually seem to remain
Isn't that
stable.
interesting?
And yet you have some that are actually moving forward, and you have some inside the blood vessels. And I wonder what the difference is between each one of those cells. Why isn't it a bulk population? Obviously, there's different cells there.
Exactly, because they're
So you have the
heterogeneous.
opportunity of then doing expression profiling or something that characterizes those cells more
Right.
deeply.
So what we've started to do is some sorting of the cells based on fluorescence properties, so we can distinguish between, for example, the endothelial cells and the cancer cells, obviously based on fluorescence. But also, in order to examine those cells that are moving, you have to by hand take them out. And so we have started to do that and do some expression profiling. And then I showed you the data for the metastatic cells, but we just did some very simple expression data based on what the reviewer wanted us to do. So only do as much as the reviewer says, right? But we do have a lot of other data and a lot of other very interesting data about how the cells behave as far as expression of the different EGF receptors, the VEGF receptors, whether they're in 3D or 2D or co-cultured or not cultured, whether they're mixed in with the cells or just one cell layered on top of the other and different. And it's all different. All the expression patterns are different. And so, again, it starts to make you think about all of the other data that have been done in 2D. How meaningful was that? Because when you examine cells in 3D like this, in these more natural soft tissue environments, they just behave differently. Cancer cells grow a lot slower, for example. Like everything is on a slower timeline, and the expression is just different. Yes.
Angela, how many different cancer cell types have you tried this with?
Yeah.
And are there differences in their behavior in this model?
So we've done this with lung cancer, prostate cancer, glioblastoma, melanoma, and we have the osteosarcoma. We just haven't done that one yet. But we have the cells. So anybody have any other interesting kinds that we'd be interested to try?
And similarities and differences
Very
in terms of their behavior?
Some of them migrate faster than others. Yeah, and some of them probably, I imagine, is going to require some other factors too, mediators, chemotactic mediators too. Yes. Any questions from anybody else off-site? Feel free. Go ahead.
Have you done this with any patient samples actually, or just cell lines? Because we noticed when we try to put a lot of patient samples into mice, they won't actually take.
Yeah, so that's a very good point. The ones I've shown you here are just cell lines, but we are working with a dermatologist, actually a pathologist, Alessio Giubilino, who's been looking at different melanomas, from aggressive and non-aggressive melanomas from patients. And that's a study that we actually currently have brains funding from the Cancer Center or medical school. Who does those, the brains? The Cancer Center, the med school? Med school, yeah. So we're currently looking at that.
Have you thought about looking at kind of the opposite point of migration, like tumor infiltrating lymphocytes, ways to get product into the tumor bed as opposed to looking at it coming out?
Yeah, exactly. Yeah, very good. So in fact, we have started incorporating cells of the immune system into this, and started to see, for example, NK cells that can go towards the tumor. We just haven't done enough of those studies yet. So yeah, some more funding would be good too. But yeah, it's a very good point, and that's something that we're working on right now. Yeah, T cells and NK cells. Any more questions?
Any other
from
questions
anyone?
or comments? All right. Thank you
Okay,
very
well,
much.
thanks, everyone.